we have the two cell types here first according to their function. If you try to examine the body that we have all over our body, there are two cell types according to their function. The first of these cell type is called your somatic cells or body cells. Okay, so somatic cells or body cells. These are the types of cells that we have that are not involved in reproduction. Okay, so that means your liver cells, the cells in your stomach, the nerve cells, the ones in your brain, your skin cells. These are all types of somatic cells or body cells because they are not in any way involved in reproduction. And these types of cells are reproduced through mitosis. Okay, so remember we have two types of M phase. Your somatic cells, all types of your cells in your body that are not involved in reproduction, these are all formed through mitosis, okay, through PMAT, through prophase, metaphase, anaphase, telophase. The second type of cell that we have here are your gametes or your sex cells, okay, so sex cells or gametes. And these, of course, are used in reproduction. And all these types of cells, basically, there are just two sex cells that we have. For humans, of course, that's the egg cell and the sperm cell. We call these the gametes. And these are reproduced or these are produced through meiosis. Okay, so when you are looking at your two types of M phase, remember you have your inner phase, you have your M phase, and your cytokinesis as the three stages of your cell cycle. For your mitosis, okay, mitosis as type of your M phase, all types of your body cells, your somatic cells, all the rest of the cells in your body that are not involved in reproduction are formed during mitosis. But for your sex cells, your egg cell and your sperm cell, they are formed through meiosis. Okay, so that's one of the differences between your mitosis and meiosis. Now, your gametes, of course, I've already mentioned, you have your sperm cells for the males, and of course, you have your egg cells, these for the females. They are formed through gametogenesis or meiosis. For sperm cells, we call that spermatogenesis, okay, making of your sperm cells. For egg cells, we call this oogenesis. Okay, making of your egg cells, that would be called oogenesis. So again, these are the different types of gametes that we have as part of our sex cells. Okay, now we also have two different types of cells according to the number of chromosomes. Okay, according to the number of chromosomes, you have your diploid cell and your haploid cell. Now, when you say diploid cell, this is represented by 2N. Okay, so when you say diploid or 2N, this means this cell has the complete set of chromosomes. Okay, it has a complete set of chromosomes. In humans, complete number of chromosomes that we have would be 46. Okay, there's 46 chromosomes in humans. So if your cell has 46 chromosomes, that means that is a diploid cell. Okay, it has a complete set of chromosomes that you have in your body. Now, somatic cells, body cells, those cells that are not involved in reproduction, those cells that are produced during mitosis, those are actually diploid cells. Okay, so those are diploid cells. Now, the other term is your haploid cell. When you say haploid cell, on the other hand, this is represented by just N. That means that's your 2N divided by 2, so you only have N here. When you say haploid, this only has half the number of chromosomes. Okay, so it only has half the number of chromosomes. So in humans, when you say haploid cells, this will only have 23 chromosomes because, as I've mentioned, as humans, we have 46 chromosomes in our body. Okay, all your body cells would have 46 chromosomes, except for your sex cells, which are haploid. That means they only have 23 chromosomes in them. Okay, so your sex cells, those cells that are produced during meiosis, they're only haploid cells. That means they only have half the number of chromosomes, which actually makes sense. So if you have the egg cell of the mom, which has 23 chromosomes, and you have the sperm cell of the dad, which also has 23 chromosomes, because it is also haploid, it's also a sex cell. If they fuse, if they have fertilization, if your sperm cell fertilizes the egg cell, forming a zygote, the zygote now, the baby, is going to have 46 chromosomes. Okay, so that's 23 coming from the egg cell, and of course also 23 coming from the sperm cell. Okay, so us as humans, we all have 46 chromosomes. Okay, except for our sex cells, of course, they're haploid, they only have 23 chromosomes. Okay, any questions so far before I go to the next slide? Any questions before I go to the next slide? Okay, so again, you have your body cells or your somatic cells, all types of cells that do not are not involved during reproduction or for reproduction, and they are formed during mitosis. They're all diploid. Okay, they all have the complete set of chromosomes. For humans, they have 46 chromosomes. You also have your sex cells or your gametes. Okay, so these cells are, of course, involved in reproduction. They're formed during meiosis, and they are haploid. That means they only have half the number of chromosomes. In humans, that would only be 23 chromosomes. Okay, if you have no questions, I'm going to move to the next slide. Now, the other terms that we have here are the first term that you have would be homologous chromosomes. Okay, homologous chromosomes. These are some of the terms that you are going to see in our discussion of meiosis later. Now, when you say homologous chromosomes, this is a set of one maternal and one paternal chromosome that pair up with each other during meiosis. Okay, and you also have the term tetrad or tetrad. This is a group of 4 closely associated chromatids of a homologous pair. So if you have a pair of chromosomes here, you'd call this your homologous chromosomes. The blue color one, this one here, is coming from your father. The red color one comes from your mom. Okay, so if they pair up during meiosis, you'd call them homologous chromosomes. Okay, so you'd call these homologous chromosomes. Sometimes you just use the term homologs. Okay, so homologs or homologous chromosomes. And you'd also call them a tetrad. Okay, when you say tetrad, of course, that means 4. And as you can see here, they actually have 4 chromatids. And so they are also termed tetrad. Okay, again, we are going to see these terms. We are going to talk about these terms as we talk about meiosis later. So again, this is a pair of homologous chromosomes, one coming from the father, the other coming from the mother, and they are also called tetrad because they have 4 sister chromatids. Okay, if you try to count the number of strands that they have, this is 1, that's 2, this is 3, that is 4. Okay, so that means it's also a tetrad. That means they have 4 sister chromatids, and they're also homologs or homologous chromosomes because they both have the same location of their centromeres, okay? The same size, one coming from the father, the other one coming from the mom. Okay, now other terms that we have here are the term synapsis. The first one is synapsis. This happens when your homologous chromosomes pair with their counterparts and remain bound due to crossing over. Okay, when you say crossing over here, this is the exchange of genetic material. Again, the term crossing over would mean the exchange of genetic material. This happens during meiosis. I'll be explaining that later. Now, you also have the term chiasmata. Okay, chiasmata is singular, chiasma is plural. This is where the chromosomes touch each other and exchange genes. Okay, so again, we've talked about homologous chromosomes. When your homologous chromosomes would pair up, you'd call that event, or you'd call that process, the pairing up of your homologous chromosomes, you'd call that process synapsis. Okay, so that's synapsis. Now your homologous chromosomes here, again, they're pairing up. You'd call that synapsis. Once they have exchanged genetic information, they've swapped some parts of their chromosomes, you'd call this crossing over. Okay, you'd call that crossing over. This is where the exchange of genetic material happens, or sometimes we also call this genetic recombination. This is exactly the reason why you and your siblings, even if you have the same set of parents, your mom and your dad are the same, you don't exactly look the same. Okay, you're not really identical. There might be some similar characteristics. Your eyes might be the same, your nose might be the same, but you don't look exactly the same, even if you have the same set of parents, the same mom, the same dad, the same chromosomes, because of the reason for this would be the crossing over. Okay, there's genetic recombination, there's the exchange of genetic information that happens during meiosis. Now your chiasmata or chiasma, this is the location, this is the point where your genetic recombination happens. Okay, so this is where crossing over would happen between your homologous chromosomes. Okay, any questions so far? Questions? Okay, so again, when you say homologous chromosomes, these are a pair of genes or a pair of chromosomes that are the same size. One of these would be coming from your father. In our illustration here, that's the blue colored chromosome. And one would be coming from your mom. When they come together, when they pair up, you'd call this synapsis. Once they have paired up, there's going to be a crossing over. That's the exchange of genetic material. Okay, you'd also call this genetic recombination. And that point where they have the exchange of genetic information, you'd call that point chiasmata or chiasma. This is the reason why you don't look exactly the same as your sibling. Okay, even if you have the same parent, the same mom, the same dad, the same set of parents, you don't look exactly the same. Okay, you might say you're more handsome than your brother, or you might say you're prettier than your sister. Of course, you won't say you're uglier, right? Okay, so again, that's because of synapsis, that's because of crossing over. Okay, now crossing over here is one reason why we have variation. Okay, when you say variation, there's differences in your genes. Okay, one reason why you don't look exactly the same as your siblings. But crossing over is just one cause of variation. We also have different other causes of variation. The second type that we have here would be the random assortment of chromosomes. I mentioned this during the metaphase. I told you during the alignment of your chromosomes, they are done at random. That means you don't know which chromosome is going to be at the center. You don't know which chromosome is going to be found at the sides of your cell. Okay, it's done at random. Now, another type of cause, okay, another cause for your variation would be non-disjunction. Sometimes, remember, during anaphase, your chromatids, the chromosomes are going to be split. Sometimes it doesn't happen, or sometimes it doesn't happen normally. So you'd call this non-disjunction. Non-disjunction, they don't split normally. Okay, and this would cause disorders like trisomy 21, or what we call mongoloids. Okay, so this little guy here, this actually is a person. This is a boy with trisomy 21. Okay, we'd sometimes call him a mongoloid. Okay, he's suffering from trisomy 21. That means your chromosome 21 has 3 copies instead of having just 2 copies. Okay, so if you are looking at the karyotype here, a chart of the different chromosomes that you have in a person according to their size. Okay, so they're arranged according to their size. You can see that his chromosome 21 has 3 copies instead of having just 2 copies. Okay, so you'd call this trisomy 21. That means that non-disjunction happened. Something happened, and abnormality happened during your division, okay, during the cell division. Now, another thing, of course, that would cause variation is fertilization. You all know that you are all winners, okay? Because, of course, only one sperm cell can fertilize an egg cell. Only one sperm cell coming from the father can fertilize the egg cell of your mom, okay? So that means that sperm cell is the winner, okay? That was the winner. We don't know which sperm cell is going to fertilize the egg cell, okay? So fertilization is also a cause of variation, okay? So again, these are the different causes of variation that we have. Crossing over, which happens during meiosis, specifically prophase 1 of meiosis. We'll get to the different stages of meiosis after this. You also have random assortment of chromosomes, which happens during metaphase. Okay, so this one happens during metaphase, during the alignment of your chromosomes. Non-disjunction during anaphase, when the chromosomes split, sometimes some abnormalities may happen, and so this can cause disorders like trisomy 21 here. So this little guy has trisomy 21. And of course, fertilization is also a cause of variation. And we don't know which sperm cell is going to fertilize the egg cell of the mother. Okay, now going back to your meiosis, remember we said your cell cycle has the inner phase, then you have the M phase, then you have cytokinesis. Okay, now we've talked about your M phase as mitosis. Right now we're going to be talking about your meiosis. This is another type of M phase. Okay, now your meiosis, unlike your mitosis, which is done after one round of PMAT. Okay, so after prophase, metaphase, anaphase, telophase, mitosis is done. In your meiosis, it actually has 2 rounds of division. Okay, so there's 2 rounds of division. We call this, the first round, we'd call that meiosis 1. And the second round, we'd call this meiosis 2. Now, the stages are the same as the stages that you have in mitosis. Okay, it's also p-mapped. The difference again is that you have prophase 1, metaphase 1, anaphase 1, telophase 1 as part of your meiosis 1. But then again, meiosis has another round of division. Okay, which is of course meiosis 2. And it still has prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, let's go to the different stages here so that you'll know what the differences are. Okay, now remember your prophase 1 here, this is longer than the prophase that you have in your mitosis. Very important events that happens here in your prophase 1 is crossing over. Okay, we've talked about crossing over. Your homologous chromosomes would come together, you'd call that synapsis. Then they'll have recombination of genes. You'd call that crossing over. Okay, so crossing over happens during prophase 1. Now, metaphase 1 here, metaphase 1, just like your metaphase in mitosis, your chromosomes are aligned in the equator of your cell. The chromosomes get aligned at the equator of your cell. But unlike your mitosis, where you see the chromosomes aligned singly, okay, one by one, in this case here, your chromosomes are aligned as tetrad. Okay, we've talked about tetrads. When you say tetrads, that means a pair of homologous chromosomes. Okay, so here, they are aligned as tetrads. They are still paired up. Okay, they are still paired up. They're still doing synapses. Now, during anaphase 1, remember anaphase in mitosis means splitting of your chromosomes into sister chromatids. In anaphase 1 here, there's no splitting of your sister chromatids yet. There's no splitting of your sister chromatids. What happens is that your homologous chromosomes get to be divided. Okay, or they are pulled apart from each other. That means this is sometimes called desynapsis. That means they are pulled apart from each other. Your homologous chromosomes, those pairs of chromosomes that you have are pulled apart from each other. That's anaphase 1. Again, anaphase 1 in meiosis 1, there's no splitting of chromatids yet. Okay, so it's not the same as anaphase in your mitosis. Now, at the end, after telophase 1 or during telophase 1 in your meiosis 1, you come up or you've already produced 2 daughter cells. But the difference here, if you try to compare this with your 2 daughter cells in your mitosis, your 2 daughter cells here are actually haploid. Okay, they are haploid. That means they only have half the number of chromosomes. If you started with a cell with 4 chromosomes, your 2 daughter cells here now would only have 2 chromosomes each. Okay, so again, we say they are haploid. They only have half the number of chromosomes. Now again, your meiosis does not stop here. Meiosis would undergo another round of cell division. It would undergo meiosis 2, and meiosis 2 is very similar to your mitosis. Okay, very similar to mitosis. That means during metaphase 1, your chromosomes here are aligned singly. Okay, one by one, no more pairs, just like your metaphase 1 here. Okay, metaphase 1, they are aligned as tetrad. That means there's pairs of homologous chromosomes. In metaphase 2, just like your metaphase in mitosis, they are aligned singly. Okay, now anaphase 2 is the same as anaphase in your mitosis. Your sister chromatids now are going to be split. So at the end, during telophase 2, or after telophase 2, you are going to form 4 daughter cells, and all of these daughter cells are going to be haploid. Okay, so these are some differences between your mitosis and your meiosis. Your mitosis would only have 1 round of cell division. Your meiosis would have 2 rounds of cell division. There's meiosis 1 and your meiosis 2. If you try to compare the different stages that you have here, remember in metaphase 2 or metaphase 1, your chromosomes are aligned as tetrads. Okay, they are aligned not singly, just like metaphase in your mitosis, but they are aligned as tetrads. In your anaphase 1, another difference, they are not split into sister chromatids, but your homologs, your homologous chromosomes, are just pulled apart from each other. Okay, your telophase 1, unlike your mitosis, where you have 2 daughter cells that are diploid, they have the complete set of chromosomes. In here, you have 2 daughter cells, but these daughter cells are haploid. They only have half the number of chromosomes. Now, after this, meiosis is going to undergo another round of division. And this division now, meiosis 2, is very similar to your mitosis, Okay, so metaphase, your chromosomes are aligned singly. Your anaphase, they are going to be split into sister chromatids, but at the end, after meiosis, you have 4 daughter cells, and all of these daughter cells are haploid. Okay, and remember, these daughter cells here are your sac cells, the sperm cells and the egg cell. Any questions before I go to the next slide? Okay, now if you try to compare meiosis and mitosis, you might see this in your benchmark on Thursday. Okay, there are similarities. We start with the similarities first. They both undergo DNA replication. That means they both undergo the S phase of your interphase. They both create new cells, or which we call daughter cells. They both occur in plants and animals. They always start with one parent cell. Okay, so these are the things that are similar with both mitosis and meiosis. Now, mitosis, again, you end up with just 2 daughter cells, only 2 daughter cells. And this is undergone by your somatic cells. When you say somatic cells, again, these are body cells. These are the cells that we have in our bodies that are not involved in reproduction. Okay, this would create body cells like your liver cell, blood cell, muscle cell. The purpose of your mitosis is for growth and maintenance, okay, growth and repair. This would result with the same number of chromosomes as parent cell. That means in mitosis, your daughter cells are going to be diploid. You start with a diploid parent cell, you end up with 2 daughter cells that are also diploid. They have the same number of chromosomes as the parent cell. For humans, that is going to be 46 chromosomes. Okay, so it creates a human cell with 46 chromosomes, creates 2 diploid daughter cell. Now, on the other hand, your meiosis, this would end up with 4 daughter cells. Okay, instead of 2 for mitosis, you'll have 4 daughter cells. This is undergone by germ cells. When we're talking about germs here, we're not talking about bacteria. We're not talking about cells that would give us diseases. Okay, the germ cells that we're talking about here are the stem cells in your embryo. Okay, so these are undergone by your sex cells, your gametes, and of course, this would create more sex cells, your sperm cell and your egg cell. Okay, so your gametes, your sex cells would undergo meiosis instead of mitosis. The purpose, of course, for this would be your reproduction. And of course, this is also for recombination of genes for variation. As I've mentioned, your phase is not exactly the same as the phase of your brother, your sister, even if you have the same set of parents, because of meiosis, okay, because of crossing over, which happens during prophase 1. And this would result with the result would be half the number of chromosomes as a parent cell. It would create a human cell with 23 chromosomes. These are your sex cells. It creates 4 haploid daughter cells. So again, if you start with your diploid or with your parent cell having 46 chromosomes for your humans, you end up with 4 daughter cells, 4 instead of just 2 for mitosis. But all of these daughter cells are going to be haploid. That means they only have half the number of chromosomes. If we are talking about humans, then they're only going to have 23 chromosomes instead of having 46 chromosomes, which actually, of course, is correct, because if you have a sex cell with 23 chromosomes and it gets to fertilize an egg cell also with 23 chromosomes, that is going to be 23 plus 23. You have a zygote, you have a baby with 46 chromosomes total.